In this video, we will discuss about the in vivo results of Siglec-10 capping. So, there is one challenge that they faced while doing the in vivo results of the Siglec-10 capping. One of the challenges is that Siglec-10 is not expressed in the mice. However, its homologous Siglec-G is expressed in the mice. So, in order to do this experiment, they did it in two different manners. So we will go through these two different experiments one by one. So in the first experiment, what they did is they inject first these ASPC-1 PDAC cells, and it has the this Siglec-10 ligands, and after like 10 days they inject this human monocyte derived macrophages. It has the Siglec-10 that is expressed on them, and then treat them with isotype control and anti-Siglec-10 antibodies. And what they are able to see in this case, with the anti-Siglec-10 antibodies, they are able to see a suppression in the tumor volume compared to the isotype control case. And these things they have quantified on the right hand side of the panel B, where it is clear that the decrease in tumor volume is statistically significant as p-value is 0.0003. Thereafter, they did check the transcriptomic analysis of this macrophages, and they are able to find out that these immune activation pathways, survival pathways, MAP kinases, AP1 genes involved in these pathways are upregulated in the Siglec-10 antibodies. It can be seen here. Along with that, the phagocytosis and cytoskeletal remodeling genes are also upregulated, and the metabolism changes is clearly evident in the Siglec-10 antibodies treated macrophages compared to the isotype control. So this in all together indicates that there is a activations and increased phagocytosis of the macrophages when they are treated with Siglec-10, or when they are treated with Siglec-10 antibodies, or when their Siglec-10 is been capped so that the Siglec-10 cannot be interacted with the ASPC PDAC cells. So once validating this, they next move on to the different next set of experiment, or make the system more biological compatible. So in that case, what they did is they first knock down the homologous Siglec-G from the mice and knock in the Siglec-10 in the mice. And these things they have confirmed using the flow cytometer analysis of the different immune cells that is shown in the panel F, where we can see that the Siglec-10 knock-in mouse shows and the Siglec-G knock-in mouse, Siglec-10 knock-in mouse, and the Siglec-G knockout mouse shows the expected signals for the B cells, for the DC cells, monocytes, macrophages. So they did like 4 different immune cells to confirm their knock-in and knockout, and for the control where there is no knock-in shows completely different or opposite signals, which is also expected. So once validating that they have a successful Siglec-10 expressions, Siglec-10 knock-in and Siglec-G knockout, they did in this study, they took this 2838C3 cells, which is highly metastatic in nature, in the mice, and then treat them with similar conditions, that is isotype control and anti-Siglec-10 antibodies. And in this case also they are able to see that there is a reductions in the tumor suppressions when the case where anti-Siglec-10 antibodies are used. And these things also they have quantified on the right hand side of the panel H, where it is clear that anti-Siglec-10 antibodies results in the tumor suppressions or enhanced immune response against the tumors. And thereafter they also check whether it will cause any toxic effect on the mice or not, and for that they check the body weight of the mice, and they are not able to find out a significant body weight change between the isotype control and anti-Siglec-10 antibodies. So all these results and the previous results, they made a conclusion that this Siglec-10 could be a potential therapeutic target that can be used to enhance immune response against the against the pancreatic tumors, and at the same time it will also suppress the tumor proliferations that is evident from both of these in vivo studies. So this is what they finally conclude, that Siglec-10 could be a potential immunotherapeutic target to suppress the tumor growth. I hope you understood how they come up to this point, and what are the results that they have shown in the main figures that we have gone through number of videos. We have gone through one by one in our videos. If you need to understand more about these experiments or this paper in detail along with the supplemental figures, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.